Welcome to Labster. You are about to start the gene linkage simulation. Here you will learn how to determine if two genes are located close together on a chromosome and how to use this information to assess whether a woman is likely a carrier for specific BRCA gene mutations that increase the chances of developing breast cancer. This simulation, using the lab, perfect. Rosa was recently diagnosed with breast cancer, and now she is admitted to the hospital. Dr. Cruz, who is in charge of her treatment, thinks she potentially has hereditary breast cancer. Rosa's daughter, Geraldine, would like to know this information ahead of time.
Hi, I am the genetic counselor who is attending Rosa and her daughter Geraldine. We'll begin by analyzing Rosa's DNA and determine if she has a BRCA1 or BRCA2 gene mutation using linkage analysis. People with mutations in these genes will have a risk for developing breast cancer 5 times higher than the general population. Linkage analysis helps us determine if the trait is inherited in the family, as alleles that are located in close proximity to each other on the same chromosome are more likely to be inherited together during meiosis. Let's begin by creating a pedigree on the basis of Geraldine's family tree. A pedigree is a visual representation of a family trait documented over several generations. In this case, the trait is cancer. Because Geraldine is the individual that seeks medical attention regarding this trait, she is the proband. We have received more information regarding this family. Apparently, several family members suffer from. Geraldine also mentioned that her grandmother died of breast cancer when she was still in kindergarten.
Before we move back to the lab, I have some questions for you. Spot on. What does a diagonal line over the symbol mean? That's right. When the subject is deceased, it is represented as a diagonal line over the symbol.
Great job, you are clearly familiar with pedigrees now. To find the underlying mutation for
Before you can work in the laboratory, you need to make sure that you are dressed properly. Wear a clean lab coat, a clean pair of gloves, put on a lab coat. Go to the PCR workbench to start your experiment. Great! Now find the glove box and put on a pair to prevent contamination. Today, we will perform a polymerase chain reaction followed by gel electrophoresis. After, we can analyze the pedigree trees together. They are all very powerful tools to uncover gene linkages. To help us identify the genes of interest, we will use polymorphic microsatellite markers that have known DNA locations. As I mentioned before, during the chromosomal crossover, closely located genes will be inherited together. We can identify the location of the gene by detecting a specific microsatellite allele that is co-inherited with the defective gene. Sounds exciting, right? Before we get started, I have some questions for you. What is a microsatellite? You're doing a great job. One common example of a microsatellite is a CA repeat, where the number of repeats varies between alleles. Microsatellite have a higher mutation rate than other neutral regions of DNA, and these mutations frequently occur by slippage during DNA replication or during recombination in meiosis. Due to the higher variability, microsatellite are better markers than SNP, which have a lower mutation rate. We need to include a control in the experiment. Who would be the ideal healthy control in linkage analysis? Good thinking. We would like to compare the microsatellite alleles to determine which one is linked to the mutated BRCA gene that causes breast cancer. In order to do this, we need to study the allelic pattern within the family and analyze if the mutated allele is inherited together with the Amazing, you are ready to start the experiment. Well done. As you can see, we are using blood as a source of human genomic DNA. We received one blood sample from the patient, Rosa, and one from the healthy control, Rosa's sister, who doesn't have breast cancer. I have extracted the DNA of these two blood samples. Click on one of the tubes that contain a master mix. Great! Today, we want to analyze 4 different microsatellites using 4 different primer pairs in the samples from Rosa and her sister, and prepare a master mix for each set. I will prepare the PCR for you today, but before I do, can you tell me the purpose of PCR? Correct. We use PCR. Please click on the PCR machine to end the program. Great. The PCR. Do you remember which equipment helps us visualize the PCR products? Please click on it. Well spotted. In a lab, we use gel electrophoresis to separate The gel electrophoresis result is ready. Let's have a look at it together. The patient has 2 bands for the D17S855 marker. What does this mean for the patient? Impressive. Microsatellite are repeated DNA sequences, and the number of for the microsatellite marker D13S1699, there is only one band corresponding to one PCR product. Which word describes both the patient and the healthy control? Great work. All the women suffering from breast cancer in this family have the pattern for D17S1323, while none of the healthy women. Both the markers D13S1699 and D13S1701 are located proximal to the BRCA2 gene. Are they linked to the disease? You did it again. There is no difference between the patient and the healthy control for both. What about the markers D17S855 and D17S1323 that are intragenic to the BRCA1 gene? Are they linked to the hereditary breast cancer in this family? That's correct. In both of the markers, there is a different pattern between the patient and the healthy control. However, we need to check the genotypes of the remaining family members. Using the pedigree information, can you determine which marker is linked to the hereditary breast cancer in this family? Well done. All the women suffering from great work. Let me ask you one last question. Thank you for your score has been saved together with your 100% progress.